Hey guys, the following is an interview with Professor Mike Levin. He's a fantastic human being, fantastic thinker. Um, we only had an hour, unfortunately, and but hopefully we can meet again at some point and dive a little bit deeper into some of these fascinating areas. So I hope you enjoy the video and speak soon. Bye. Hey Mike, thanks for joining us.
Yeah, nice to see you. How are you?
Um, you're probably one of the most sort of world's most important scientists at the moment. Your work is just sort of looks groundbreaking to me. It's shocking in some places. So I wondered if we could kind of start by catching some of our viewers that you haven't seen some of your work before. If we could catch them up a bit. But I'll actually before that, could you give us a bit of a brief intro about yourself and how you ended up being, the developmental biologist, how you got here?
Yeah. Well, from an extremely early time point, I was fascinated with both technology and life. So as a kid, my dad used to take the back off of our ancient TV set, and I would look through at the vacuum tubes and everything, and just this idea that somebody knew how to put them all together in the right way, right? That was transformative for me, and that's something that I really wanted to learn. And then after that, electronics engineering. But eventually, I got my hands on a computer, was interested in programming software. At the same time, I was always interested in the outdoors. I liked spending time outside. I had a friend who introduced me to the joys of bugs and insects and things like this. And so just playing those things off of each other, and looking at living things that really have a lot of intelligence in terms of pursuing specific goals, and they have preferences and both the body and the mind, right? So you see an egg, a caterpillar egg, and then you see that it turns into this amazing organism with behavior, and then it turns into something else with different form and behavior, and how that is the same or different than the technology we have. So these are the things I've always been interested in. I was a programmer first. I got 2 degrees, one in undergrad, one in computer science, and one in biology. I then decided I had a software company briefly, but I left it to go do grad school in biology. Yeah, and ever since then, I've wanted to do this kind of research, with developmental biology being the central thing, because there you have that whole journey right in front of your eyes. You have that whole journey from physics to mind. You start life as this little blob of chemistry, and then eventually you have this organism that, in the case of a human, will even make claims about not being a machine, and all of these interesting things, right? So how did you get here? How did that process go? Yeah, that's what I'm interested in.
Cool, man. Could we kind of sort of catch people up there? So those viewers that haven't encountered your fantastic work, could we say maybe it sort of last sort of 2 or 3 years worth? If you could just sort of squeeze that in, say 10 minutes, sort of talk about the work on Xenobots, planaria, sort of frogs and the skin cells. There's some of their surprising behavior, and I know you've got a couple of slides here as well that you could potentially share with us.
Yeah, I have a few things to show. And I just want to be really clear. It isn't just my work. So I have many people in my lab right now who are working with me and producing some of these amazing things, right? So it's not just me. But the thing that we focus on is embodied mind. So we are really, if even, I mean, so my lab does lots of different things. We do things in AI and in birth defects and cancer and regenerative biology and behavior science and things like that. But all of these things are fundamentally designed to understand intelligence, embodied intelligence in different spaces, and really unconventional types of intelligence. So how do all kinds of things that we're not used to, how do they solve problems? What worlds do they live in? And so, okay, I will show you a couple of slides here. Let's see. So this is the kind of thing that both evolutionary biology and developmental biology teaches, is that around this standard thing that we think of as a human, going back in evolutionary time, back to a unicellular state, but in developmental time, of course, again going back to the egg, there's this continuum of slow and gradual change. So we really need to do a lot better than old, really stale binary categories of what's a man, machine, organism, robot. These categories are really no good. They were never any good, but now we know they're no good, because it's quite obvious that there's a continuum here. There's another continuum where we can and the community is making in technological and biological changes to these forms. And so we really need to understand the scaling of minds as they emerged slowly and gradually, right, in this continuum, and what else is possible by introducing new engineered kind of constructs at all levels of organization. So what we're really interested in is this kind of thing. We are all collective intelligences. We are made of things like this. This is the agential material that we're made of. This is a single cell, and it doesn't have a brain. It doesn't have a nervous system, but it's very competent in its local kinds of little tiny goals that it pursues. And it turns out that even this is made of an agential material too, because the molecular networks inside of those cells themselves have memory capacity. They can be trained. They can do Pavlovian conditioning and various other kinds of things. So there are many scales, and this is where what my lab works on, is understanding the intelligence at all of these different scales of organization, from the molecular networks to the cells to the tissues, organs, of course, whole organisms. And so over the last few years, we've had some pretty neat advances in understanding what kind of learning can happen in molecular networks. And the trainability of cells. We've shown examples of biological systems navigating creatively and intelligently navigating other spaces. So the space of possible physiological states, the space of possible anatomical shapes, solving problems, things they've never seen before. And that leads to questions like how do cells cooperate to reliably build a particular kind of body despite various perturbations and all kinds of changed circumstances that they've never seen before? And we've even discovered that, in fact, this goes up to higher levels where embryos work together. And I'll just show you one example here. This is one of my favorite examples. And so what you're seeing here, and this is actually rather old data now by Danny Adams in my lab, where she found the electric face. And this is part of our long-standing attempt to understand how to communicate to the collective intelligence of non-neural cells. Turns out they use electricity just like neurons do. It binds them together, and we can talk about how that works, but it binds them together into a collective whole that has goals and preferences and memories that no individual cell has. And so we've learned to read some of these memories. And so you can see what's happening here. This is a time lapse of an embryo putting its face together. This is one frame out of that video. This is called the electric face, because you can actually see, if you track the electrical patterns here, you can see this is the future plan of the face. Here's where the eye is going to go. The other eye comes later.
So Mike, it's like that bioelectric field is telling the cells where to build the face, right?
That's exactly right. In the same where and how and what shape the face should be, in exactly the same sense as the electrical properties of your neurons are telling your body what to do to execute behavior. I mean, that's exactly the same thing. So something in any kind of collective intelligence, you need a mechanism and a set of policies for the pieces to communicate with each other to create this collective. And in both of these cases, it's an electrical network. And yeah, the electrical network sets the shape, the size, the location, many of the properties of the anatomy. And so you can see that here. You can see the cells communicating with each other here to form this face. Here is watch this kind of wave. This is Angela Tung, a PhD student in my group, and a number of other collaborators studied the ability of embryos to communicate with each other. And they do this again. They form a collective, which we call a hyperembryo, which has its own unique gene expression patterns. Large groups of embryos are better able to survive various kinds of interventions than small groups and individuals. Again, this notion of a collective intelligence. And this is in terms of this bioelectricity, one of the things that you want when you find a new intelligence, one of the things you want is to communicate with it. And in order to communicate with it, you need to understand what's the interface, how does information get between you and it? And then you need to crack the code. You need to understand how to say the things that you want to say. And so how do we communicate with this collective intelligence? Well, here's an example. Suppose here's a frog embryo. And suppose we want to tell this frog embryo that, you should really have an eye back here on your gut. Okay? Of course, normally they don't have an eye on their gut. So what you can do is by looking at this and saying, okay, here's the bioelectrical pattern that says build an eye here. We have developed techniques to introduce that electrical pattern somewhere else. And if you do that, these cells go ahead and they build an eye. And so they build an eye. The eye has the same lens, retina, optic nerve, all that good stuff. And the other thing is, if you only hit a few cells here, the blue ones are the ones we injected with our ion channel. What they will do is recruit all the other ones in their vicinity to participate with them. So
does the eye still work, Mike?
So it does work. We have other data where we induce. We're using a slightly different method. We induce eyes on the tail, and we show that they can, in fact, see. I'll show you an example of that right here. This is the work of Doug Blackiston, another amazing scientist in my group. And so here's your tadpole. No primary eyes here, but there's an eye on its tail. And these eyes, they make an optic nerve here. You can see it. It does not go to the brain. It goes to the spinal cord, or sometimes to the gut, or sometimes nowhere at all. But yet these animals can see perfectly well. And we've built this machine to test and train them on visual cues. And
so Mike, just briefly, so even though that eye is kind of out of, sorry to interrupt, but even though that's out of synchrony with the target morphology, the surrounding cells still find a way to be able to integrate that eye within the surrounding tissues and so on. That's incredible, right?
It absolutely is incredible because this animal for millions of years has the brain has expected visual input from these two locations up here, and all of a sudden, in one generation with no evolutionary selection, no modifications, suddenly the sensory motor system is the architecture is completely different, and no problem. The brain takes this information that might show up on the spinal cord, and it can act accordingly. I can talk a little bit about that and how I think it works, because I think it's important. You want to should we do that?
Yes, please. That'd be great. Yeah.
Okay. I think fundamentally there's something very important here, which is that evolution doesn't make specific solutions to specific problems, which is often how people think about evolution, that, there's a froggy kind of environment and there's a particular thing that's fit for that environment, and eventually selection will give you something that works there. I actually think that's not what's going on with evolution. I think what evolution is actually doing is making problem-solving agents. It's making cognitive systems, not necessarily brains, but it's making all kinds of systems that can solve problems, because fundamentally, evolution knows that everything is going to change. Not only the environment is going to change, but also your own inner parts are going to change, because there's going to be mutation. There's going to be all kinds of processes. And this is why we see numerous amazing examples of how with the same genome, you get all kinds of different solutions, both anatomically and behaviorally, to different problems. And one way to understand why that is, and this is the body and the brain and the mind are affected equally, is to think about this kind of interesting example. So here are these caterpillars, and they have a particular brain suitable for their two-dimensional life, crawling around on leaves, and they eat these leaves. And then they have to turn into this, which is a completely different kind of creature that lives in a three-dimensional world and drinks nectar. So in order to do that, the brain is basically refactored. Most of the cells are killed off, the connections are broken, and it builds a new brain. Except that the amazing thing, and this was shown by a number of people, including, again, Doug Blackiston, before he joined my lab, that the memories, you can train these caterpillars, and the memories that they form are still present in the butterfly or moth that results. Now, one amazing thing is that, well, where are these memories? If the brain is getting taken apart, where are these memories? But there's actually an even more amazing thing here, which is that actually storing these precise memories, it would be no good, because the memory of crawling with a soft body kind of controller, crawling towards leaves for a particular stimuli is of eating leaves, is of no use to the butterfly, because the butterfly doesn't move that way. It doesn't care about leaves. It wants nectar. So what's happening here is that it's not just the fidelity of the information that's being stored here. It's a generalization and a salience that is being remapped onto a completely new body. So this kind of shape, this bow tie architecture, where This one happens to be from a machine learning thing that a machine learning architecture that people often use, where there's a lot of different kinds of data and experiences and things that come in. They get squeezed down into this bottleneck, this like bow tie architecture. They get squeezed down and generalized into this bottleneck, and then the right side of it has to reinflate and readapt this information onto new circumstances, and this happens everywhere. So for example, in evolution, this would be an egg. Organisms don't make other organisms; they make eggs, and the egg now has the difficult job of, in a new environment, maybe with new changes, maybe with mutations, who knows what's going on, that it has to construct a body that may or may not be quite the same as it was before. The same thing happens in memory. You and I, we're not caterpillar and butterfly, but still, we don't have access to the past. We have access to the memory engrams that were formed by the past that are in our bodies and minds and brains. So what that means is, at any given moment, we have to creatively interpret those engrams to build a coherent story of what am I, what was my past, what am I doing, what are my goals. This is a constant construction process, right? And there's a lot of interpretation and creativity going on here, because from this squeezed down generalized information, you can't reconstruct exactly what its meaning was. You have to, it's a constant process of sense making, of your world and of yourself. And this is all over the place in biology. The chemical networks, bioelectric networks, biomechanical networks, they all have these little bow tie nodes in the middle, which means that the rest of it, once you get your information, the rest of it is a very active, creative, sense-making process. And this is why biology is so unbelievably good at adapting to novel circumstances, because it didn't necessarily assume that things were going to go the right way in the first place. Unlike our, right, like in our computer technology, the game is make sure that the data stay what they are, make sure that nothing changes, and make sure that the bottom layers are extremely stable and there is no errors that are visible to the higher levels of programming. Biology takes the exact opposite approach. It assumes that the hardware is unreliable. You don't know what's going to happen with your hardware, right? And so then what you need are algorithms to creatively interpret your information. So that's why that I think is why the plasticity,
Yeah, I think one aspect of your work that sort of captures that beautifully is these frog skin cells. And you might just think that a frog skin, left to their own devices, they just wouldn't do much. They just form a kind of monodimensional lattice or something and just be skin cells. But could you talk a little bit about these entities in a kind of unique environment, and how actually they formed Xenobots, didn't they? I mean, this is the foundation of your Xenobot program.
Yeah, I'm gonna try. Let me see if I have those slides here. Yeah. So here's what happens. So you're looking at this thing, and it's this little self-motile creature that can take a turn in the maze and then turn around when it wants to, and so on.
Why did it turn around? Why do you think it turned around there?
We don't know. We have no idea as of yet. We have some technology that we're starting to use that will help us answer that question, but we have absolutely no idea as of yet. The thing that is important about this this construct is that it's just skin. It's basically skin cells taken off of a frog embryo, and what we try to do is to ask the following question. Typically, when you're looking at any kind of a plant or animal or something like that, and you ask, why does it look the way it looks? Why does it behave the way it behaves? There's a standard evolutionary story here, which is that, well, eons of selection. It was selected to have these specific properties. So we were interested in the question of, where do goals come from when there is no long history of selection? So novel collective beings that didn't exist before, where do their goals come from? Okay, and so we created the first thing we created were these Xenobots. This is, as I said, skin cells from a frog embryo, allowed to sort of reboot their multicellularity and make something new. And so you can see here, they have all kinds of behaviors. They can go in circles. They can patrol back and forth. They have these kind of collective behaviors here. One of the most amazing things they do is called kinematic replication. And so this, by the way, the biology here is also the work of Doug Blackiston, and the computer science part of it was done by Sam Kriegman in Josh Bongard's lab. What you can see is that if you provide them with the material, these white things are loose skin cells. What they will do is they run around and they sort of compact them into these little spheres. And because they're dealing with an agential material, these are not passive pebbles. This is an agential material. These little blobs then become the next generation of Xenobots. And guess what they do? They run around and they build the next generation of Xenobots and so on. And so what's interesting here evolutionarily is, look, you can ask, okay,
the new animals, these are new creatures, aren't they? These have never existed before.
Yes, and all of this, one of the cool things about this kind of thing is that it blows up all the binary categories that we used to have. So in many ways, yes, these are new creatures, but also not, in the sense that the cells of which they are made certainly did exist before, because they were part of an embryo and they have an evolutionary lineage as part of an embryo. But as an organism, it never existed before, and there was no selection to be a good Xenobot or anything else. People also argue, is this, we call them Xenobots. Xenopus laevis is the name of the frog, and we said it's a biorobotics platform. So some people say, well, it's not a robot, it's a living thing. And some people say, well, obviously it's a robot like all other chemical systems, including cells and us. And the problem with either of those statements is that it assumes a binary distinction. So this is, I think, pretty important. I think all of these terms, right? So organism, robot, machine, intelligent, cognitive, all of these things, I don't think they are claims about a specific object. I think they're descriptions of how we intend to relate to that object. So when you say something is a robot, what that means is that you're going to try to capitalize on its controllability. You're going to try to program it. You're going to try to get it to do something useful. That is how you intend to relate to it. And this is certainly a biorobotics platform, because it can be used to do all kinds of useful things in the environment. But at the same time, there's a different stance you could take on it, which is much more of an organismic stance, to say, never mind us trying to program it, what does it want to do, and what are the capacities that it has? And both of those things are true at the same time. We don't have to pick one or the other. It's how are you going to relate to it? So look, this is the thing about this, is that these are completely normal cells. There are no scaffolds here. There's no synthetic circuits. There's no weird nanomaterials. It's all we've done is engineered by subtraction. We've liberated these cells up here from the instructions of these other cells down here, which normally bully them into having a boring two-dimensional life as the outer skin of a tadpole. So when we take them away from all that, now you get to really find out what they do. This is what they want to do left to their own devices. What they do when they're not being controlled by other cells is this: they turn into a Xenobot. Then they have some crazy developmental sequence. This is an 84-day-old Xenobot. It's turning into something. I have no idea what it's turning into. It's having some kind of developmental stage. So and then it has different behaviors. And what we don't know yet, well, we know some, but it's not published yet, is, for example, their cognitive properties. Can they learn? They can learn some things. We're still studying that. And a couple of interesting things. So when you ask, what does the frog genome know from its eons of experience in the world? Well, reliably, it makes this. So what it makes is hardware that under normal circumstances does this kind of thing, but it also is able to do many other things. The exact same hardware is able to do many other things. For example, this. And in fact, they express hundreds of genes that frog embryos normally don't express. So they're able to dip into their genome to pull out new kinds of new kinds of proteins that might be more important for their new lifestyle. And that's the one of the significance of these kinds of synthetic things, is because they're showing us that what life actually makes is hardware that has multiple different behavioral modes.
Yeah. I mean, it sort of feels like we went through a phase where, we thought that DNA was going to explain absolutely everything. There were, you were reading articles about behavior being mapped, sort of traced to certain DNA information and so on and so forth. But I think we're now saying that the instructions for where to put organs and the chirality and so on and so forth is just simply not contained in the DNA, is it? So I was just going to ask, where is that information stored, Mike?
Yeah. Well, a couple of interesting things, and then I want to come back to actually the Anthrobots, which are kind of a next step beyond the Xenobots. So with the question of where is it, let's just take a look at this example here, and then I can talk a little bit about how to think about those kinds of things. So look, here's a flatworm, planarian. These are amazing creatures, among other things. They are incredibly regenerative, so you can cut them into many, many pieces and they regenerate. Every piece builds a nice new worm. They are cancer resistant. They are immortal, so there's no aging in the system. And they are incredibly resistant to transgenesis, meaning putting new genes in there. People have been trying to make transgenic worms for about 40 years now, and to no avail. And there's an interesting reason for that we can talk about, which is that the thing about planaria is that they do their damnedest to basically ignore the genome and any kind of defects in it. They are committed to, because of their unusual lifestyle where they accumulate mutations all the time, they are basically committed to the idea that the hardware is going to be unreliable, and they pretty much ignore it. And one of the things about planaria is that, unlike every other creature, you can call a stock center and you can get a fruit fly with curly wings, or you can get a mouse with a crooked tail. There are different color eyes. There are lines of genetic, mutants. There are no lines in planaria except for our two-headed form here. This two-headed line of planaria, and that's not genetic. And I'm going to show you how we did it. So here's a normal planaria, and you chop off the head and the tail, and this middle fragment will reliably give you a nice one-headed worm. So we asked this question: how does the fragment know how many heads and how many tails it should have? Okay, and so what you can see here is that there's this interesting voltage gradient in this animal that says one head, one tail. And what we went and we're able to do is to change that gradient and say, no, actually, you should have two heads. It's kind of a mess still, because this technology is really hard, especially in planaria. It's very hard, but you can see basically what's happening here. There are this now encodes two heads. Now, what's really important, so this is what you're looking at here, is a bioelectrical pattern. We are literally reading the mind of the collective intelligence of cells. That this is showing us the representation of what it thinks a correct planarian looks like. I'm using all these cognitive terms very specifically because not only functionally do they fit here, due to the system's navigation of anatomical space, which is just like animals navigating three-dimensional space, but all the molecular components are exactly the same. We steal everything from neuroscience. All the tools, the reagents, the concepts, everything maps over perfectly, because neuroscience is not just about neurons. So it goes much deeper than that. And so what we're able to do is rewrite this pattern memory of what a correct planarian looks like. So we communicate with this collective intelligence by using drugs that open and close certain ion channels. So that's our language. And we know just a few words in this language. And one of the things we know is how to convert a pattern that says we should have one head into a pattern that says, no, we should have two heads. Now, what happens next is if you cut this animal, you in fact get a two-headed animal, because you've changed the representation of what a normal planarian is supposed to look like. And what's really important here is that this voltage map is not a map of this two-headed body. It's a map of this perfectly normal looking one-headed body. It's just that until you cut, it looks like this. The anatomy is normal. The molecular biology of it is normal. Only the memory of what it's going to do if it gets injured in the future. That's pretty wild. That's a counterfactual memory. That's the beginning of kind of mental time travel, where certain animals can imagine things that are not true right now. Yeah, so things that are going to be, or things that were in the past. But the somatic intelligence can do it too. And this is a future goal of what I'm going to do if I get injured at some point in the future. And you can see it, and we can rewrite it. So a single body of a planarian can store at least, I'm sure, more, but we've nailed down two representations of what it thinks a correct planarian should look like. And so if you do this, you get these two-headed worms. The reason I keep calling it a memory is because it has all the properties of memory. If you take these two-headed worms and now you cut off the primary head, you cut off the ectopic secondary head, you don't do anything else, you don't treat them in any other way, they will forevermore continue to generate two-headed animals. So now we get back to the original question. Okay, so where is it stored, right? Where is the number of heads stored? Well, in an important sense, it's not in the genetics, because these animals have exactly the same genetics as the original animal. There's nothing wrong here. We have not edited the genome. So you could imagine if we toss this in the Charles River here, a couple of, 100 years later, some scientists come along, they scoop up some samples, they say, oh, a two-headed form and a one-headed form, cool, let's sequence the genome and find where the speciation event happened, right? They're not going to find anything, because that is not where it is. So in an important sense, the information is not in the genome. But here's the part that is in the genome, which is that what the genome is doing is encoding the microscopic hardware that every cell gets to have. So the proteins, it's not encoding the number of heads, it's encoding the hardware that every cell gets to have. And that hardware, the cool thing about that hardware is that by default, it reliably has a certain functionality, which is it generates an electric pattern that says one head. It just so happens that it's rewritable. That pattern is rewritable on the fly. We should not be surprised by this at all. I mean, a lot of times people hate this sort of computer analogies in biology, but the part where it's good is this notion of software and physiology as software. So the genome nails down the biological hardware. And like all hardware, you turn on the juice, it has some kind of baseline behavior that it's going to do by itself. But then you find out that it's actually really cool hardware. It's reprogrammable. And experiences that it has in the future will change how it behaves. And it has a memory, and it has problem solving, and it has all kinds of other things. So biological hardware is amazing. And yes, you get some default patterns from the emergent properties of the hardware, of the genetically specified hardware, but all kinds of things can happen after that. And there are many amazing examples.
Mike, could I just ask? So those chemicals you were using with the planaria to affect the bioelectric field, is that the beginning of a sort of an interface that would support your anatomical compiler?
Yeah, precisely. So in planaria, we have to use chemicals, as I said, because they are completely resistant to other ways of doing it. We also use other kind of strategies to, for example, in frog and human cells, we in culture we use other kind of strategies. But yeah, it's the interface that the anatomical compiler is going to be using to program novel goals into these cells. Now, I have no doubt that as time goes on, other interfaces will be added. So lots of people are working on biochemical signals, biomechanical signals. There's some cool work in ultra weak photon emission, so optical signals between cells. Who knows what else? There may be a million other things. But the bioelectric interface is really powerful because that's one of the key. It's the protocognitive medium that the cells are using to store their collective memories, and not individual cell memories, although maybe with we don't know about that, but the collective memories of the group are stored in this electrical in the electrical states.
That's really, really interesting. Do you think? I wonder if we've got a little bit of time to talk about that. The collaborating embryos.
Yes.
Is that Angela Tung's work?
Correct. Yep.
Yeah.
That's
because there's something absolutely astonishing about that. These embryos local to each other actually collaborating in some sense. I'd love to hear more about that.
Yeah. Well, let's see. I'll tell you the findings, and then we could talk about what the implications are. The basic finding is that if you take a large group of embryos and a small group, or even a single embryo, and you expose them all to some kind of a teratogen, something that impairs their ability to complete normal development. That might be a chemical, it might be a genetic defect, it might be there's lots of different things. It doesn't have to be a drug, but there are many things. What we found is that larger groups deal with it way better than small groups or singletons. And so there's some kind of, and of course we did all the things that, scaling up the drug correctly and all of that, right? So we took care of all of that. It's quite remarkable that there's some kind of a group problem solving effect. And one of the most interesting things we found is that every member of the group has to have been impacted by the problem in order to help fix it. We originally thought this is very counterintuitive. We originally thought that, okay, so here was our original model. We thought, well, what's happening here is that you get hit by this teratogen. You're not quite sure how to complete your anatomical journey, but because some of the damage is a little bit is stochastic, it's noisy. This embryo might not know how to complete one aspect of it, this other embryo a different aspect, but if they just talk to each other together, they have all the information they need. It's like the old wisdom of the crowds thing where people try to guess how many jelly beans in a big jar. Individuals are way off, but the collective is usually spot on to the number, right? So we thought it was something like this, but that way of thinking about it makes a very specific prediction, which is that if you mix so a large group in this case is about 300 between 200 and 300 embryos. If you were to mix, half of that group would be embryos that got exposed to the teratogen, and half of that group would be pristine embryos that never saw the teratogen. You might think that that would be fantastic because the pristine ones, they're not confused about how to do things, and they would inform the others, and then then everything would be great. What we found is that doesn't work. That doesn't cause the effect. If all of the members had to have been affected by the teratogen in order for this to happen. So it's actually something much more subtle, and we have some hypotheses about what's going on. But it's something about all of the collectives trying to resist the same kind of problem. And what we then found is that you can actually watch them talking to each other. Now, in this case, just to make it easier for the video, this is just done by mechanical damage. There's a little needle that hits this center embryo, and you can see all of these other guys find out what happened to him within seconds. They're spreading information. They're spreading information all around. And then what we did was we did some transcriptomics to ask, okay, so do large groups express different genes than small groups? And what we found is that, first of all, even without any perturbation, large groups of embryos express some different genes than small groups do.
Wow.
But which is wild and unexpected. But also, if you do perturb them in some way, large collectives that are able to do this, we call that CEMA, CEMA, cross embryo morphogenetic assistance. This CEMA effect, while the group is doing the CEMA effect, it expresses a whole bunch of new genes that small groups which can't do the CEMA effect, that they don't express. So what we have here is a weird thing. I called it a hyperembryo because, in a certain sense, there is a collective here that's more than the sum of its parts. It has communication, it has gene expression, and it has morphogenetic competencies that its parts don't have. It's a new level of organization. Now you might think that's pretty weird. We're looking at a collection of embryos and we're giving it a name. But think about what any individual embryo is. When you look at what we call an embryo, here I will I have a slide on this too. Yeah, here we go. So look, this is an early blastoderm. Let's say it's an avian, bird embryo or a human or something, and any kind of amniote. You look at this, and then there's, I don't know, 50,000 cells or something, and then you say, oh, look, there's an embryo. Well, what are you counting as one when you say there's one embryo? I mean, there's 50,000 cells, and there's tons of stuff inside of each cell. What is there one of? Well, what there's one of is something very interesting. There's a commitment to one particular story, is what it is. It's an alignment of a goal of navigating anatomical space to exactly this kind of outcome. All the cells here are committed to doing exactly this. That's what you're counting when you say it's an embryo. So already the embryo already is a collective. And this is true because what you can do, and I used to do this as a grad student with duck embryos, you can take a little needle and you can temporarily put some scratches into that blastoderm like this. And what happens is each one of these islands doesn't feel the rest. And so it self-organizes an embryo, and then eventually they heal, and you get these twins or triplets or quadruplets. And so talk about genetics, right? How many individuals are there in an embryo? Well, you don't know, do you? It could be anywhere from 0 to half a dozen, depending on what happens next. It's an emergent process, right? It's a self-organizing process. It isn't nailed down by the genetics, although the genetics give you this amazing hardware that's capable of doing that. And then every embryo, one of the things it has to do is it has to figure out, where do I end and the outside world begin, right? Because every cell is some other cell's neighbor. So in a case like this, well, where am I, and where's where do my borders end? And this becomes a very important thing, both cognitively and morphogenetically. And this idea of being able to figure out how many individuals there are in some kind of excitable medium like this is huge for cognitive science too. Just think, if you didn't know what a brain was, and somebody showed it to, or what a human was, and somebody showed you this brain and said, hey, how many people do you think fit in here? Like, how many personalities do you think could fit in here? We have absolutely no idea, because we don't understand the density, right, of how much, what can a cubic centimeter of brain tissue support in terms of individuals. And so all of this stuff with split brain patients and dissociative identity disorders and all that tells you that yeah, in all of these, this is a dynamic process
Yeah,
of
yeah.
setting right the border between self and world.
Just wanted to ask a quick question on that, actually, Mike. I'm been thinking about the sort of Portuguese man of war lately, and that's an example of a sort of collective intelligence that's made up of actually what are regarded as different species as well. So there's a bunch of different animals cooperating to form that what looks like a sort of singular collective. But is there some distinction between that kind of collective agency and the
Yeah.
collective agency that you have in, say, a single, say, human embryo? Is there a difference between those two? Or should we just actually regard ourselves as part of the continuum? And we're not really an individual. I mean, there's animals inside my body that don't even have my DNA, you know?
Yeah.
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 So what are your thoughts on that?
Yeah, I was looking for a slide. I don't. It's not in this. It's not in this set of slides. But I'll describe it. Look. First of all, we are all absolutely collective intelligences. We're all made of parts. In fact, I think all intelligence is collective intelligence, because I can't imagine how you could have an intelligence that isn't made of
Yeah.
parts.
Yeah.
So, right, if you're not made of parts, then you can't change, you can't learn, you can't have cognition.
And that's what they're figuring out in artificial intelligence, isn't it? That's part of the the revolution there as well. Yeah.
I think it will be. Yeah. So the thing about, but this is I think important. There's an old approach to this, which is kind of the animist approach, which says minds everywhere. Every rock has a spirit in it. Every ecosystem is a being with goals and what, right? So there's that approach, and then there's the more recent sort of scientific kind of teleophobia, where people say no, there aren't minds anywhere, and some of them say there aren't even we don't have them. It's kind of eliminative kind of strategy, right?
And illusionism, yeah.
Yeah, so I think both of those are fundamentally wrong, because they treat this question as a philosophical question that you can just settle by making a decision. The sun is a mighty spirit. I mean, maybe,
yeah,
but I
prove
don't.
it.
Yeah. I don't think any of this can be settled just by deciding it. I think you have to do experiments, and so the way it works is, and then this gets back to your question about the Portuguese man of war and all that. What you have to do is you have to hypothesize some kind of problem space that your agent is working in. You have to hypothesize what you think its goals are, and you have to hypothesize what competencies does it have to reach those goals. Meaning, what is the level of intelligence? William James said intelligence is the ability to get to the same goal by different means. Like, how much creativity and problem solving can you muster to solve your goals, right? Once you do that, then we can, and somebody else will do a competing, we'll have a competing view. So we look at a brain and somebody says, I think that's a fantastic paperweight, and the best thing it's going to do is just go down with gravity, and that's it. All it knows how to do is follow gravitational gradients. And someone else looks at it, no, you've missed the whole point. It does all this other stuff in behavioral space, and then in linguistic space, and so on. So the thing about making these hypotheses is that you do perturbative experiments. You put barriers between it and its goal in whatever space you think it's working on, and you get to find out how your particular framing worked out for you. Did it? The point is not anything goes, and you can just sort of seek intelligence anywhere. So people say to me sometimes, well, you've shown memory in these gene regulatory networks. They're just a bunch of chemicals. You might as well say the weather is cognitive. I said, well, I don't know. Have you tried training it? I actually have no idea. You could do. I certainly wouldn't say it is or isn't until somebody does the experiments of setting up some kind of, I don't know, you could look for habituation, sensitization in weather patterns. I have no idea, but it's an empirical question. It's not, you can't just decide these things. So for the human and the jellyfish, I don't know what competencies it has. I've never worked with it. Somebody ought to figure out some hypotheses about what it might be doing, and then you will find out what the degree of collectivity is that it has. Maybe it's just a collection of loose parts that really don't make an individual at all. I don't know, but these are empirical questions. I want to show you. It's a question that actually gets to.
Just briefly, Mike, there seems to be a sense in which we just can't help classify things, and that can be quite unhelpful sometimes. There seems to be there's just this general human tendency to say, well, here's a speciesation and things like that, and really, you're pointing out, no, there's a continua there.
There's a couple of examples of this in others. So for example, there's this notion of an adult. Okay, so now, so what's an adult? You know, we all know nothing magic happens to you on your 18th
You
birthday.
can vote.
It's
Yeah.
yeah, but it's a total fiction that's used because it makes it lubricates this, you know, various social structures, and it makes those things easier.
Mm.
Um, we know it's a continuum, but for the purposes of specific activities, we pretend there's a sharp. We need
Mm.
to understand that all these other categories are just like that. They
Yeah.
can be useful for certain cases, but let's not
Mm.
pretend they're real. And
Mm.
but they do have important implications. They have implications for ethics, and they have
Yeah.
implications the way that look at you know, the electromagnetic spectrum, right? Everybody understands that it's a continuum, but there are different things you can do with gamma rays than what you can do with
Mm.
long, you know, electromagnetic, you know,
Yeah.
ELF waves. And depending on how you look at it, it facilitates what kind of relationship you're going to have with it,
Mm.
what kind of technology, what kind of. So I think it's important not to classify, as in pretend that there are these sharp categories, but to develop rational, defensible, interaction protocols for how are we going to ethically relate to beings that are very different from us. I mean, this is critical. We are going to
Yeah.
be
Yeah.
surrounded by never mind humans that are non-neurotypical, but there are going to be cyborgs and hybrids and chimeras and things you've never seen before, things evolution has never seen before. Our old ways of deciding how we're going to relate to things has to change, and we have
Yeah.
to
Yeah.
expand our notion of compassion to other beings,
Mm.
just because our finding it hard to imagine what it's like to be them is not a good a way to, you know, to cut off moral concern.
Yeah.
So
Yeah.
I think, you know, I think it's very important, and even biomedically, like, look, this is how we started life, as tiny little cognitive light cones that basically only cared about very local goals, right? This thing has some metabolic goals and some proliferative goals around about the size of a single cell. Well, during evolution, you get this incredible, and of course, the bioelectric networks are a huge part of how that scales. You get this incredible scaling of the cognitive light cone, where the group of cells, this is a salamander limb that when you cut it off, it rebuilds and then they stop.
Mm.
Right,
Mm.
the group of cells has this grandiose construction project goal. Now, how do you know it's a goal? Because when you
Mm.
deviate it from this, it gets right back up and it does
And how does it
exactly
know
that.
where to stop? How does it know where to stop? Like, where is
Bingo,
that?
bingo.
Where is
That's
that
it.
blueprint? Where is the blueprint?
Well, we don't know in particular for the salamander limb, but I showed it to you for the face and for
You
the
did.
planaria.
Yeah.
It actually, right, it actually has a bioelectric memory of exactly what it's going to build, and
Mm.
when it finishes building it, that's when it stops. Neuroscientists who try to do neural decoding and, you know, sort of decode the goals of living, you know, mice and zebrafish and humans and whatnot. So, but the key here is that the goals of this thing have become immense, right? Individual
Yeah.
cells don't know what a limb is. They don't know what a finger is. They still have tiny little goals, but the collective and this giant electrical network has a memory of, hey, we're building this thing, and if anything goes wrong with it, we're going to try to fix it. That is a massive expansion of the boundary between self and world, right? This creature has a tiny little boundary. This
Mm.
creature has a much bigger boundary. But that process has a failure mode. That failure mode is cancer. So what will happen is that when these cells become electrically disconnected, they revert back to this state. So this is human glioblastoma. This is a video by Juanita Matthews, another scientist in my group. She studies the bioelectrics of cancer, and this is glioblastoma. And these cells. So here's what happens when we inject a human oncogene into a tadpole. The cells electrically immediately disconnect from their neighbors, and that border between self and world now shrinks.
Mm.
It becomes very small. The cells are not more selfish than they were before. They're exactly as selfish, but the self is now smaller. They just have smaller selves. So what happens is that now all they do is they pursue their local tiny little unicellular goals, and the rest of the body to them is just outside
Mm.
environment as far as they're concerned.
So, Mike, could we say that what appears to us as a single entity, take myself, right? Am I a collection? Am I a super boundary of these kind of embedded mini boundaries? Is that what I am?
Very yes, yes.
And would you be prepared to call it a Markovian boundary?
I think the Markovian kind of paradigm is a good start for looking at these things. I think that, yeah, I think we are absolutely a nested overlapping set of competing, cooperating selves.
Yeah. Yeah.
I
So,
think it's an interesting question. If you
mm,
know, if aliens were to come down, which level of organization would they want to communicate with? It's not
mm,
super clear. I think it would have a lot to do with what their cognitive frame rate was. In other words, if they operate on million-year kind of
yeah.
life cycles,
Specious present. Yeah. Yeah.
they might be to them only the ecosystem might be an interesting agent to talk
Mm,
to. It's not clear,
yeah.
but
Yeah.
again, all of these things are true at once. So right now, you and I are having a conversation, and we are communicating at the level of this large emergent cognitive self. But in biomedicine, we would like to have conversations with organs and with tissues, and with
Mm,
and try to get them to do things. So
yeah. Yeah.
and we know what it's like to communicate with even larger things than humans. You might get a letter from the town that you live in. It's not a letter from any one person. It's
Mm,
literally you're being informed by the town, and you'll write something in reply, and you might
yeah.
be
Yeah.
happy about it or not. So
Mm,
we have these. It's all about understanding that there are multiple levels, and it's in the eye of the beholder what level you think you're
mm.
talking to and why, and what your
It,
tools are.
it seems to be a case of compete or collaborate, essentially, at all levels. And when I was talking to Carl Friston the other day, his view is that this agency, if you like, and this sort of it goes just all the way down, and it just never stops, probably. And he thinks equally it probably goes all the way out and never stops that direction as well. But I just
Yeah.
had just one question for you, and I think you've already touched on this. I'm just conscious of time. I know you're insanely busy, Mike.
Sure.
So
No, please.
again, I really appreciate this.
Sure.
Um, so hang on, let me just think that through. So it's this idea of collective intelligences, and maybe scale up to something like the Gaia hypothesis and say, is there a singular kind of self-awareness, maybe even with sort of metacognition? I suppose my question is, how could we test that? And I think you have touched on this with the weather and so on and so forth, but how would we be able to test that singular that sort of metacognition?
Yeah. Great question. So if you think about well, a few things. First of all, there is, I think, as far as I know, there's no guarantee that larger scale groups are going to be more intelligent than the pieces they're made of. Sometimes that's true, but I don't think that's guaranteed at all. So we have to be open to the idea that the larger groups we're made of might have different levels of cognition, different types of intelligence, and different goals. This is pretty much a existential project for humanity, because we make these collective intelligences all the time. Internet of Things, political structures, social structures, financial structures. What these things are going to want, we have no idea. What capacity to achieve their goals do they have? We really have a very, very poor science of understanding this. So how do you know when you're part of a larger collective intelligence? I mean, for smaller and simpler things, we can do experiments, and in fact, I've written papers on exactly how to do protocols for exactly how to do it, certain ways to check for habituation, association, anticipation, problem solving. But let's assume for the moment that we're asking the question, how do we know when we're part of a larger collective intelligence? I think this is really hard. I think that probably there's some sort of Gödel type theorem that says that you can never definitively know that you're part of a collective. That's my guess. I can't prove it, but that's my guess. But I bet you can get evidence for it. And one way to start thinking about this, so in one of the blog posts where I talk specifically about this question of how do you know if you're part of a larger intelligence, I have this imagined conversation between two neurons inside a brain. And one neuron says to the other, yeah, the universe is this cold, unfeeling, mechanical place. It doesn't care about us. And then his friend, the mystic, neuron says, I can't prove it, but I have this weird feeling that there's a grand design here, and there's some kind of pattern. And I think our universe wants something. And the first one says, you're crazy, and he says, no, stuff just happens according to the laws of chemistry, And the other one says, no, but these waves of back propagation and these things that are happening, I feel like they want something. I feel like there's more to this. And so can we prove it? Probably not. My conjecture in that blog post was that what it would look like to us is what Jung and Pauli call synchronicity. I think that's what it would look like. It would look like something it would look like something that at our level has no order or explanation for why these two discrete events were happening.
But the
But
symbology might still guide us in some way. So it
I think
might be
so.
a kind of an instruction from that superstructure, if you like. Yeah, that's
I think
fascinating. That's really fascinating.
that's what I think it would look like to us. It would look like synchronicity, something that
Mm,
has salience, that has importance to us mentally, but the physical facts have nothing in common with each other.
mm. Yeah, that's really interesting. Um, I think we're quite close to the end now, but there was one thing that I just wanted to pick up with you, and we'd had a couple of email exchanges. I don't know whether you remember, but I raised the question of the homunculi thing with you, and I mean we were talking about sort of personal identity and continuing forms. And I said, what is it? What is that thing that's doing the observing? And can I just quote what you said? What you said to me? It's quite
Please,
interesting.
I don't remember. Please go
All
ahead.
right.
I hope I
but you,
still believe it, but tell me.
but it's absolutely fascinating, and I'm still sort of scratching my head about it a bit now, Mike. And it says, my suspicion is that we agents are thoughts that have closed the loop in Hofstadter's sense, and are now reinforcing our own persistence through time and spawning off thoughts of our own. It's a scaling along a continuum virtualization stack, ranging from extremely simple thoughts to advanced multi-scale thinkers. And then you went on to say it bottoms out in the active inference of particles. So there are homunculi aplenty, but it's not an infinite regress. That's foundational. So I mean, that's really deep. Could you expand on that a bit?
Well, I'll try to do that in the next 5 minutes.
Yeah.
And by the way, and I'm happy to meet up again. But the thing
Oh,
is,
cheers, man.
this is very recent stuff. Okay, so this is like right at the edge of. So I'm by no means am I trying to say that this is a worked out theory or anything like that. But here's how I'm thinking about it. I'm just going to start with a very and there's a paper I just put up a preprint about it, so you can kind of see the whole thing.
Oh yeah, and
But
by the way, that you've got a fantastic academic site as well on YouTube as well, haven't you? So I'll stick the link into that in the info. But yeah, carry
thanks.
on.
Yeah. So I'm just going to start with a very quick science fiction story. So imagine these creatures come out of the center of the earth. They are lava, kind of. They come from the core. They're incredibly dense. They use gamma ray vision because they're just so dense. So they come up here, and everything on the surface is gas to them. It's gas phase. They can barely see us. It's a tenuous plasma to them. It's not real. It's just like it's so rarefied. But they can, they know it's there because they have instruments and they're scientists. And one of them says, I've been watching this gas, this tenuous plasma that surrounds our planet, and there's patterns in this gas, and these patterns kind of look like they're doing something, and they kind of look agential. And
it's us.
and right. And the other one says, well, that's crazy. Patterns in a medium can't do anything. We're real. We're actual agents. You can't. And he says, no, I think. And he says, well, how long do these patterns last? And he says, well, about 100 years. He said, well, that's nonsense. What can you do in 100 years? We live for millions of years. Okay, so now you start to understand that the distinction between patterns in an excitable medium and aka thoughts, so contents of the cognitive mind, aka versus the actual thinker, that's a little bit in the eye of the beholder. So what I'm playing with now is this notion that there is no binary distinction between thoughts and thinkers. I'm not the first person to say this. William James said thoughts are thinkers. I'm not sure if this is what he meant, but that's what kind of set me down this road. So he probably anticipated this, like he did with a lot of other things. So once you understand that maybe there is no harsh boundary between thoughts and thinkers, what you can imagine is in an excitable cognitive medium, there might be simple patterns that exist and dissipate quickly. There might be more complex patterns that are a little bit self-reinforcing. And of course, people have studied, hypercycles and metabolics and in the environment for many decades, right? So patterns that are self-supporting. And then you might have something that's even more interesting, which is patterns that can spawn off other patterns. We've seen this too. You see this in the game of life. You see this in all kinds of immediate. So if you're willing to accept a continuum of cognition, then maybe what we're saying is that really good selves, really good minds, are ones that can not only support their own existence, but actually spawn off others and actually have thoughts of their own, which may or may not go on to support. So that's, this is all pretty airy fairy stuff, but I
that's
think
almost a necessary condition for the persistence of an identity in a way, though, isn't it?
I think it is, because the whole point is that these patterns have to be they have to be active, because passive, as we talked about with the memories, passive memories are no good in biology. And I think the thing, I don't spend a lot of time trying to define life, but I think that one thing that we call life are things that are really good at scaling up cognitive light cones and generating thought patterns that can generate other thought patterns.
Fantastic answer to
There's
that
a lot
question.
to be done there. Yeah.
There's a hell of a lot to be done, I think. And talking of which, you're at the end of the hour now, so good luck with the rest of the day. And your work's absolutely amazing. And thanks so much for joining us. And I'll
Thank you so much.
Cheers, Mike. Take care.
Yeah, that's very kind. Thank you.
See you later, man.
Yep. Bye bye.
Bye bye.